First of all, I want to thank the organizers for inviting. I really enjoyed this morning's talks, and just like every one of you, and I'm definitely learning a second language. I would characterize myself as a prostate cancer researcher with a focus on genomics. So what I'm going to talk about is pretty much in the context of prostate cancer research. Whether the findings going to be going to apply in other contexts, I'm really not sure. I guess one thing with this affiliation with Hopkins is we have a long history of frozen tissue collection. And we have in the tissue bank about 2,000 frozen tumor samples. And I actually took advantage of those. And in the past 10 years, I have been using frozen samples. So why would I get into this trouble into FFPE, paraffin blocks? For three specific reasons that has something to do with my research interest overall. First of all, I am in transition into analyzing small volume prostate tumors. We know those tumors, they were not harvestable, meaning they cannot be harvested for frozen tumor collection. The reason for that is we have to preserve the very small amount of tumor for diagnostic purposes. So we actually do not have the material to analyze those small volume tumors, which I think is really the key to address the modern clinical problem of overdiagnosis and overtreatment in prostate cancer. So that's in the clinical setting of prostate cancer. Second of all, I'm also interested in analyzing pre-malignant lesions such as high-grade PIN. And those lesions, they are very difficult even to identify in frozen tissues because the freeze-thawing process would pretty much destroy the morphology. You can't identify those lesions. So if you are able to work with FFPE, that's going to allow you to identify the molecular correlates of those defined lesions. Third, although we have tons of frozen tissues, we still need more. And if we can utilize those hundreds of thousands of specimens in the archival blocks, that would be wonderful. So those three reasons really prompt me to get into this area. So what are the problems? What's causing all those problems? So I basically define those pre-diagnostic variables and post-diagnostic variables. And those variables, some of them can be controlled after diagnosis, such as storage time. And of course, you can analyze the tissues with relatively recent fixation, and you can also analyze aged tissues. That's something you can control depending on what what your research focus is, and you can control those. And you can also control for prospective purposes, you can control storage condition. You can store them at room temp, or you can store them at -20. You can control your RNA extraction methods. There are different methods to extract them. You can also control the platform of choice. But those pre-diagnostic variables, they are hard to control. Unless you have data or you have some political cloud that you can influence the diagnostic setting, or you can influence the clinicians, those would be very difficult to control. And those, you cannot control the physician, whether he's going to operate, how he's going to operate on the patient. And there are robotic surgeries, there are open surgeries, and we do know that ischemia time is different from those two surgeries. It can cause delay in fixation based on how the tissues are processed. And this part, we actually control that very well in Hopkins, but they can always be improved. The fixation type and the fixation time and tissue processes, those are all in the pathology type of area. And we try to control them, but still it's very difficult And you have to, convince people that, even to standardize the tissue processor, it's, you have to take into consideration of the economics, how much it costs to replace the tissue processor, etc. So we try to work on one variable at a time, in the hope that by controlling, different variables, we can turn this basically, we always thought, FFPE tissues, they are so bad that we can barely use them, so they were really trash mine. So by controlling the variables, by constantly improving, we can turn that into a gold mine.